now that we've talked about prokaryote cell division, we're going to look at how eukaryotic cells divide. But before we get into too much detail, we need to remember that eukaryotic cells have a nucleus. So it causes a lot more complications and complexity than when we have just the prokaryote cells with no nucleus. So their cell division is a little more complex. So before a eukaryote cell can divide, the nucleus and other organelles must be copied. So the first step is DNA replication. So that means we're going to make a copy of each chromosome. So right here is one chromosome. But then since it's copied, we now have this second rod-shaped, and they join together at a structure in the middle right here called the centromere. And that's what holds those two chromosomes together. Once that DNA has been copied, then the nucleus needs to divide. So that means we need to divide the chromosomes in a process we call mitosis. Then, after mitosis occurs, then the division of the rest of the cell. That cytokinesis, remember, means the division of the cytoplasm. When we are done, we have two daughter cells, and they are unique. They are not the same. So remember, with prokaryotes, we had identical daughter cells. But with eukaryote cell division, we end up with two unique cells. So here's your little diagram. We start with the parent cell here. Then our DNA has replicated. Then our chromosomes line up in the middle. And eventually they pull apart since they were duplicated. And then we end up with two daughter cells. So mitosis is the division of the nucleus. And this only occurs in eukaryote cells because they are the only ones with a nucleus. And there are 4 phases of mitosis. And so you have to definitely know the order. And you need to know what happens in each of these 4 phases. So a lot of students just remember PMAT for prophase, metaphase, anaphase, and telophase. So the first phase is prophase. This is where the chromosomes break down or duplicate. Let's change that to duplicate. The nuclear membrane breaks down, and the structure called centrioles move to opposite poles, and spindle fibers form between the centrioles. Our second stage is metaphase, and this is when the spindle fibers attach to the centromeres, and those sister chromatids line up at the center of the cell. Anaphase is when the spindle fibers shorten, and they pull the sister chromatids towards opposite poles. And lastly, we have telophase, and the chromosomes will uncoil, the spindle fibers break down, and new nuclear membranes reform. So this is a little bit easier to visualize it with a picture. So let's label some parts. So right here, this structure, that is a centriole. And there's only 2 of them, and they're both at the poles. Then you can see that we've got these white dashed lines. That is the nuclear envelope. And it's dashed because it's breaking down. These spaghetti-like noodle-looking structures, those are the spindle fibers. And so in this prophase, Notice how the chromosomes have been duplicated because we have the X's. That tells you they've been duplicated. The centrioles are at the poles, and our nuclear envelope is going away. When we look at metaphase, the chromosomes, you'll notice, are lined up here in the middle. So metaphase middle. And so each of these spindle fibers are attached to the centromere of each of these chromosomes. Then, when we move into anaphase, notice how the X's have been pulled apart because the spindle fibers are shortening. So remember, these are the spindle fibers, and they're shortening. I always think of anaphase as reeling in a fishing pole. Fishing line, I guess. And so the spindle fibers have been cast out, they've hooked onto a chromosome, and now it's time to reel them back in. So these chromosomes are actually moving towards opposite poles. Then telophase, notice how we start to have that nuclear envelope, and now it's dashed again, but instead of breaking down, it's reforming. And our spindle fibers are shortened. And instead of our chromosomes, our DNA has uncoiled. So we're going to probably spend some time practicing and identifying each of these stages of mitosis.